Evening, everyone. So, this is Kanika Gupta, your TA for the session, and I'll be helping you with your doubts in the week 8's lecture. So, I hope you guys have seen the videos and you understood the concept well. Still, if there are some confusions, we can discuss them here. Okay, so I'll start with the discussion part. Are my slides visible to you all? I hope they are. So, yes, let's move to further. So, when we are now the part of the molecular techniques that have been used in molecular biology have started, and now we'll be discussing different techniques in the upcoming lectures, upcoming weeks. But in the past week, what did we studied is the blotting technique. So basically, in molecular biology, when we want to study some proteins, DNA or RNA, then we need to blot them or we need to shift them on a suitable membrane or a suitable medium through which we can specifically probe them with some kind of probes or antibodies or something, so that a specific study can be done. So for that, blotting techniques are quite useful and have been used from a very long time. So today we'll be discussing about three blotting techniques: the southern blotting, northern blotting, and western blotting. Eastern blotting, we are not going into the details, although this type of blotting is used in case of the when any carbohydrate molecule is involved. In that case, we go for eastern blotting, basically the post transcriptional modification, translational modifications. In that case, we go for eastern blotting. Apart from that, if you want to study the protein, we'll go for western blotting. If you want to study the DNA, then we will go for southern blotting. If you want to study the RNA, then it is northern blotting. So this is how these different blottings are named according to for which molecule they have been used. Okay, so this is what we have discussed now. Southern blotting for DNA, northern for RNA, western for protein, and eastern blotting is used when any carbohydrate molecule is involved. Basically, a post-translational modification is there on protein. In that case, we go for eastern blotting. So first of all, what is southern blotting? What happens in southern blotting? So southern blotting, as I've told you, is used for DNA. What are the steps while doing this kind of process? So first of all, the genomic DNA must be isolated from the cell. We should have the sample of DNA. Then the digestion to generate the fragments. Then the separation of DNA fragments on agarose gel, because in blotting we are transferring what we have separated. So they must be separated on gels that have different pore sizes, and then they must be transferred to a suitable membrane where they can be studied. Step 4 is the transfer of DNA from gel to the NC membrane. Step 5 is the hybridization with radiolabeled probe. Step 6 is the washing and autoradiogram generation. So autoradiogram is basically the result, what we see after probing and all, to know what was there in the sample. Sorry class, bear with me. So first of all, genomic DNA isolation. So you guys have studied the plasmid isolation in the previous lectures. So this is also a similar kind of process. Like lysis of cell will be done, then enzymatic digestion of that whole thing will be done, so that the proteins, the RNA part, that all can be digested. Then the precipitation of DNA from whole of that pool will be done. After that, the purification will be done. If any leftover protein or RNA is still there, then the leftover thing will be removed. Also, the DNA, as we all know, is wrapped around histone proteins. So to remove those histone proteins also, this purification with chloroform and phenol is important. Then the precipitation with absolute alcohol and solubilization in water will be done, so that the samples can further be used for digestion with different enzymes, and then separated on the gel. Okay. Second step is the generation of suitable size fragments. So we have isolated the genomic DNA. Now, as they said, digestion to generate fragments. So in this step, we will be generating the suitable size fragments from DNA. So how that will happen? That will happen either by restriction digestion or by mechanical shearing. Restriction digestion, what happens is genomic DNA can be digested with a frequent DNA cutting enzyme, and those enzymes can be EcoR1, BAM-H1 or SAU3A to generate the random sizes of DNA fragments. The criteria to choose the restriction enzyme or pair of enzymes is such a way so that a reasonable size DNA fragment will be generated. So that basically what here we want is the size of the DNA should be that much that can be easily seen on the agarose and that can be easily transferred to the membrane also, so that we can study it better. It's not like we will generate very small fragments or very large fragments. It should be a suitable size. So for that, the pairs of the enzyme that will be used to digest that DNA, that must be decided very wisely and very thoughtfully, that what size of fragments of DNA we want. Okay, so as fragments are randomly generated and are relatively big enough, it is likely that each and every genomic sequence is present in the pool. Okay. And second method, mechanical sharing. Genomic DNA can be fragmented using a basically either by sonication or by vortexing. We can also uh, make the fragments of the DNA, generate the fragments of the DNA. So while restriction digestion, what happens is generally 10 to 20 microgram of the genomic DNA is used when we are using EcoR1 overnight in the presence of appropriate buffer and BS if required. Then a small aliquot from the restriction digestion reaction is taken out and checked on 0.8% agarose gel to just to check whether the fragments have formed or not. Appearance of smear with visible bands indicate complete digestion of the genomic DNA and the suitability of the sample for southern blotting. So it will be checked on gel and then we can say whether it should be further proceeded for southern or not. Next is the separation of DNA on agarose gel that we did in the previous step also, just to check. So all of the sample will be then will be run on the agarose gel, and the separated fragments will be then transferred to the NC membrane. So here also it can be capillary transfer method, it can be other methods also. So in capillary transfer method, what will happen is soak the gel in 0.25 normal HCl for 10 minutes with shaking at room temperature. This step depurinates the DNA and facilitates the transfer of larger DNA fragments. This step can be omitted if the fragment of DNA is of smaller size. So this is an optional step. Remove the deep urination solution. Wash the gel with double distilled water. Submerge the gel into alkaline solution for 30 minutes at room temperature. With gentle shaking, and alkaline solution will denature the DNA. Now we want separate fragments and separate strands of the DNA. So to remove the denaturation solution, then again the washing will be done. Then the preparation of nitrocellulose membrane will be done, on which this DNA fragments will be transferred. Then the preparation of radio labeled props. So basically we didn't went into details of the transfer of DNA from NC membrane. You guys must have heard that in the video and must have understood also. We're just discussing an overview. So in steps, what we did, we isolated the genomic DNA, we digested the genomic DNA, then we saw them on the agarose gel. Then the membrane was prepared, the transfer of that contents of agarose gel to the membrane. And then again, now we have our sample on the membrane. Now how we will see it? So to see it, to visualize it, we need the radio labeled probe. So those radio labeled probe will be then prepared. Though it can be a random primer method in which in this method a random primer is used to anneal to the template, and then a PCR reaction is performed in the presence of radio labeled nucleotide. After PCR, newly synthesized DNA strand is labeled with radioactive nucleotide. ठीक है, तो यहाँ पे एक रेडियो लेबल्ड प्रोब को प्रिपेयर करने के लिए पहले पीसीआर से उसे एम्पलीफाई करेंगे, देन उसको रेडियो एक्टिव कंपाउंड से लेबल करेंगे, एंड देन जब हम उसको अपनी मेम्ब्रेन पे जो हमारे डीएनए है उससे जब उसके साथ जब हम हाइब्रिडाइज करने के लिए रखेंगे, प्रोब करने के लिए रखेंगे, देन दैट रेडियो लेबल विल बी यूज्ड टू स्टडी. ठीक है, सो नेक्स्ट स्टेप विल बी हाइब्रिडाइजेशन. हमने रेडियो लेबल प्रोब बना लिए. नाउ hybridization and development of the blot to see the strands. So place the membrane in a hybridization tube and pre-hybridize in 10 to 20 ml of hybridization solution at 65 degrees Celsius for at least 30 minutes. Okay? And then the probe will be prepared, and then that probe will be supplied to that hybridization tube, through which this hybridization of probe will occur with the bands already present on the southern membrane, on the NC membrane, and then that will be visualized and we'll develop it. An autoradiogram will form, and we can see different fragments of DNA, and we can tell whether the gene of interest is present there or not, how many copies of gene of interest is there, and many more things. Okay. Next is the northern blotting. So northern blotting, as I've already told you, is used for RNA. Okay, to study the RNA. So, sorry. In northern blotting, again, first of all, the mRNA isolation will be done. Total mRNA of the cell will be taken out. Then the separation of mRNA fragments on agarose gel will be done. Then the transfer of mRNA from gel to NC membrane will occur. And then that mRNA that has been transferred to the membrane, then it will be probed with the cDNA probe. First of all, the probes will be prepared, and then it will be probed with the cDNA probe, which is called as the hybridization. And then last will be washing and autoradiogram development, basically the visualization of the results. Sorry. So how the RNA will be isolated? Again, first of all, the lysis will be done. Then after lysis, release of total RNA either by lysis buffer containing detergent or by homogenization in case of hard tissue. Mixing of poly T containing beads with the total RNA preparation. Due to mutual exclusive affinity, mRNA binds to the poly T beads. Why they will bind to poly T beads? Because of the presence of the poly A tail. Okay. Now wash the beads with the washing buffer to remove non-specific cross-contaminating species. Elute the mRNA from the beads and its purity can be checked on polyacrylamide gel. RNA gels are performed under denaturing conditions. Presence of secondary structure in RNA allows the faster migration of RNA on agarose gel. So that is why they are done in a denaturing condition, so that the secondary structure, the stem structure, the hairpin loop structure, the bulge, the internal loop, and so on, these kind of secondary structures do not form, and the RNA won't migrate faster, so that we can come to know the exact size and other details about the RNA. So as a result, it gives less time for the molecule to interact with the agarose gel, and consequently less resolution within the different RNA species. So destruction of secondary structure in the RNA structure minimizes these effects and allows better separation, better resolution, better visualization. Okay. So that is why we go for a denaturing gel. Now the RNA sample and the agarose gel contains formaldehyde. Just a minute, class. Now the RNA sample and the agarose gel contains formaldehyde to denature the secondary structure present in the RNA and prevent the reformation of double-stranded regions in the RNA structure. ठीक है, तो दोबारा से स्ट्रक्चर्स को बनने भी नहीं देगा व्हेन द denaturing gel will be used for the RNA separation. ठीक है, सो प्रोसीजर अगेन नॉर्दर्न ब्लॉटिंग का प्रोसीजर क्या है? स्टेप वन इज द आइसोलेशन ऑफ mRNA, affinity purification, oligo T beads will be used. Step 2 is the preparation and casting of denaturing agarose gel so that the RNA can be separated on that and can be visualized on that. ठीक है, sorry. So basically we are not going into the again the details of the how the casting of the denaturing agarose gel will be done. You guys must have studied. Acha, one thing that you should remember here is whenever we are isolating the DNA or doing some work related to RNA, the water should be DEPSI treated. So DEPSI here is what? Diethyl pyrocarbonate. It is a strong inhibitor of RNAs. So whenever we treat the water with DEPSI, it will inhibit all of the RNAs present there, and your RNA won't be affected. It will be safe and sound. So it should be treated with DEPSI. Okay. So we are not going again into the details of the casting of the agarose gel or preparation of the RNA sample. You must have understood it by the videos only. Still there is some confusion. We can discuss it in the end. Then the loading of the RNA sample will happen on the agarose gel. When the agarose gel will be loaded, again the denaturing condition and the deposit condition should be maintained. It shouldn't be absent in at any step when we are performing some RNA work. Okay. So next will be then the staining of the agarose gel. Just a moment. Yeah, loading of the RNA sample, then the staining of the agarose gel. After staining of the agarose gel, sorry, yes, we missed some step here. So agarose gel की हमने यहाँ पे staining करी है, then after it should be transferred to the NC membrane. After transferring it to the NC membrane, so basically when we are transferring it to NC membrane, there are some things that needs to be kept in mind. The membrane should never be dry. It should be kept moist always. It should have Wattman filter paper on both of this side so that it should get some support. It shouldn't come in contact with any kind of the ions directly. Sorry, not ions. Any surface which can have an RNAs present in that directly. So for that, Wattman paper should be there. Also, the air bubbles trapped inside should be removed completely. Then after the buffer should be transferred and all of these steps. So basically the transfer to the gel of the gel to the NC membrane should be done under again the denaturing condition and the DEPSI condition. We shouldn't miss the RNAs free step at any step while we are performing the northern blotting. Okay. Then the preparation of the radio labeled probe of the DNA will be done. After the preparation of this radio labeled probe, this uh, we are again not going into the details. Then the hybridization will be done as we've done in the southern blotting case, because now we want to visualize. We have the gel, we have transferred the contents of the gel to the membrane. We have the probe also. Now we want to see the bands. We want to see where our at what size our mRNA has come, or whatever we want to study the copies of the mRNA, the prevalence of the mRNA in that particular tissue, the presence or absence of the mRNA, whether that protein is being formed at that time or not when we took out the tissue or the cell. So whatever we want to study, we can study by after visualizing it. So first of all, again the hybridization part will be done. Then the cross linking with UV rays will be done. Then after what? Then after cross linking, then we leave it for probing overnight again. And after probing, after getting hybridized the probe, next day it will be generated through autoradiogram, and we will visualize the results. and these results will actually tell us the transcriptional activity of the gene, as I've told you, at which stage, at what time, at like how many copies of mRNA. All of this information can be taken out then from the visualized results. Okay. Next is the Western blotting. So as I've told you, Western blotting is done for what? It is done for to study the proteins, size of the protein, copies of the protein, anything you want to study, you can study it from the Western blotting. So the Western blotting is a popular technique to detect the specific protein present in the crude lysate or homogenate. It uses the separation of different proteins in the gel electrophoresis, preferably SDS page, and then the transfer of proteins onto a solid support such as nitrocellulose membrane. A primary antibody directed against the particular protein of interest, and then a secondary antibody is used to detect the primary antibody and gives either colored or chemiluminescent signal. So basically, chemiluminescent signal will be detected. Uh, there can be methods for that. One is the HRP method. HRP is the horseshoe reddish peroxidase. Is basically, sorry, horseshoe peroxidase is basically an enzyme which which help in the generation of the chemiluminescence when it is provided with its particular substrate. We'll be studying that further in the lecture. So, what is electrophoresis? So, electrophoresis is an electrokinetic process which separates charged particles in a fluid using a field of electric charge. It is most often used in life science to separate protein molecules or DNA, and can be achieved through several different procedures depending upon the type and size of the molecule. A particular electric field is provided in a particular direction so that the charged molecules can move accordingly and can be separated depending upon their size. Hence, electrophoretic mobility V is directly proportional to the charge and inversely proportional to the viscosity of the medium. Okay, as much as more viscous will be the medium, less will be the mobility. Obviously, it will face more opposition, so the mobility will be less. And size and shape of the molecule. If the size is too large, also then it will face uh, a repulsion in the medium, so that it the movement will be somewhat restricted in case of the large size. And also the shape, linear shape or circular shape, depending upon that also the mobility is affected sometimes. In the case of relative mobility, it is directly related to the charge or radius of the molecule. For a globular protein, the radius of the molecule is related to the molecular mass of the macromolecules. Okay, so basically it is the charge, it is the sorry, the size, the shape, and obviously the viscosity of the medium, the charge on the protein. All of these factors decides the mobility of that particular protein in the gel, in the medium, so that they can be separated differently and then can be further studied. Okay. So reagents used in the gel electrophoresis are different buffers, and reagents that can be used are, first of all, the timid. It catalyzes the acrylamide polymerization. Basically, whatever gel you are making it with the help of the acrylamide. So that acrylamide must be polymerized. So timid helps in catalyzing that polymerization, otherwise it will take a long time. So timid helps in the catalysis of the polymerization of acrylamide, so that that liquid that we are using to form the gel can solidify and form a gel. Then ammonium persulfate APS is the initiator for the acrylamide polymerization. So the initiation of the acrylamide polymerization will be done with the help of APS, and the catalysis of this reaction will be done by timid. Then the tris HCl, it is the component of running and gel casting buffer, providing the buffering effect it provides to the ions that are present there. Then the glycine, it is the component of running buffer. It provides a charge, and also it is the guiding molecule in the uh, in the gel. So yes, next is the bromophenol blue. It is the tracking dye to monitor the progress of the gel electrophoresis. Comasi brilliant blue, it is used to stain the polyacrylamide gel. After you have ran the gel, you just want to visualize, you don't want to transfer it to the membrane, then in that case this Comasi brilliant blue will be used. Next is the sodium dodecyl sulfate. It is used to denature and provide negative charge to the protein. Next is the acrylamide monomeric unit used to prepare the gel. Bisacrylamide are the cross linkers for acrylamide. So basically the reaction, when we are saying it is the catalysis or the initiator of the acrylamide polymerization. So polymerization happens basically with the help of bisacrylamide, and for that kind of reaction this APS antimide will be used. ठीक है, so acrylamide polymerization का mechanism क्या है? APS in the presence of timid forms oxygen free radicals and induces the polymerization of acrylamide monomer to form a linear polymer. These linear polymers are interconnected by the cross linking with bisacrylamide monomer mash with the pores, and the size of the pores is controlled by the concentration of acrylamide and the amount of bisacrylamide in the gel. ठीक है, so the pore size can be differentiated depending can be different depending upon the amount of acrylamide used and the bisacrylamide used. In a vertical gel electrophoresis system, we cast two types of gel: stacking gel and resolving gel. First, the resolving gel solution is prepared and poured into the gel cassette for polymerization. A thin layer of organic solvent such as butanol or isopropanol is layered to stop the entry of oxygen and make the top layer smooth. After polymerization of the resolving gel, a stacking gel is poured and the comb is fitted into the gel for the construction of different lanes for the loading of sample. So the running of gel, the sample is prepared in the loading dye containing SDS, beta mercaptoethanol and glycerol to denature the sample, and presence of glycerol facilitates the loading of the sample in the well. Basically, when you are loading the sample in the well, then it must settle down in the well so that it won't spill off. So in that case, glycerol is very important and helpful. As the samples are filled vertically, there is a distance drift between the molecules at the top versus at the bottom. In a lab, this problem is taken care once the sample run through the stacking gel. So basically stacking gel stacks them in a single line, and after that when they are going to resolving gel, they move together. So the problem taken care, once the sample runs through the stacking gel, the pH of the stacking gel is 6.8, and at this pH, glycine is moving slowly in the front, whereas the tris HCl is moving fast. As a result, the sample gets sandwiched between the glycine and the tris and gets stacked in the form of the thin band. As I've told you, stacking will align them in the single line, and as the samples enters into the dissolving gel with a pH 8.8, the glycine is now charged. It moves fast, and now sample runs as per their molecular weight. Due to SDS, they have equal negative charge. After tracking dye reaches to the bottom of the gel, gel is taken out from the glass plate with the help of a spatula, and it is stained with the Comasi brilliant blue dye when we don't want to perform the Western, and the dye stains proteins present on the gel. Now again there are procedure for the preparation of the sample, the electrophoresis of the sample, the transfer of protein gels on the blotting membrane. So we again are not going into the details of that. Assembly of transfer cassette, I hope you guys have understood it from the lecture, so we are not going into the detail. Now once we have transferred the gel on the membrane, now we need that the membrane when we have transferred it, now we want that apart from the places where we have our desired protein or whatever protein that we put it in the gel, apart from that all other places should not catch our antibody and should not show some kind of the background signal. So to prevent that, we generally do the blocking of this western blot using 5% BSA. uh, so basically, it is uh, in BS in 5% BSA, either you can leave it for 30 minutes or 1 or 2 hours, or you can leave it overnight. It is okay then, but it should be kept at 4 degree in case you are leaving it for overnight. The blocking is done. Then after next day, the washing will be done. Excess of the BSA will be removed and the washing will be done. And after washing, the blood should be probed with the primary antibody. After probing it with the primary antibody for the nearly 1 hour, again the washing will be done. The excess of the antibody should be removed, and then it should be probed with a secondary antibody so that the detection part can be done easily. So after that, the blot development will be done. There are multiple ways to develop the blot and detect the protein present on the membrane. The different reagents available to develop western blot are the chromogenic one are the HRP based. In this, the substrate is a 4-chloronaphthol, Debsia and 4-chloronaphthol, which gets oxidized product to form a purple precipitate. DAB NiCl to forms the dark brown precipitates. TMB forms the dark purple stain. और अदर जो कंपाउंड्स हो सकते हैं दैट कैन बी यूज टू डेवलप वेस्टर्न ब्लॉट आर एल्कलाइन फॉस्फेटेज यूज हो सकता है. ल्यूमिनेसेंस के केस में HRP बेस्ड होता है ल्यूमिनॉल एंड H2O2. ऑक्सीडाइज ल्यूमिनॉल गिव्स ऑफ ब्लू लाइट and then that will be detected using particular detectors and filters. Alkaline phosphatase again can be used here also. So the colorimetric detection: wash the membrane with TBS to remove detergent. Place the membrane into the colorimetric reagent and protein bands appear in 10 to 30 minutes. Stop the reaction by washing in distilled water. Air dry the membrane and photograph for permanent record. Then after, the chemiluminescence detection is also done to further clarify the bands and the proteins. So this is how the western blotting will be done. The northern blotting and the southern blotting will be done. We haven't went into the much details of the procedure because that you guys must have understood from the video. We just had an overview of what can be the different ways to develop different kind of the molecules. Now we'll be discussing the questions from the assignment. If you guys had any issue till now, you can ask me. Otherwise, we'll be moving to the assignment part. Again, the assignment practice will go as we have been doing it for the longer time. That I'll ask you guys a questions. I'll read the question. You guys will write the answer in the chat box. No one will speak out loud the answers so that everyone can participate. Okay. So we first of all, the question number 1 will take into account. Just a moment, guys. Yes. So the question number 1: to precipitate the genomic DNA from the aqueous phase, following is commonly used. Answer to the question in the chat box. बाकी लोग बाकी लोग कोई आंसर नहीं देना चाहता. देवद्रिता के अलावा देवद्रिता. सॉरी इफ आई प्रोनाउंस योर नेम रॉन्ग. बाकी दोनों लोग आंसर नहीं करेंगे. That's okay. So the answer to question number 1 is C. Obviously, the absolute alcohol. Absolute alcohol, chilled absolute alcohol is required to isolate that. Okay, question number 2. In southern blotting, the DNA fragments are separated by size using. Question number 2 का answer. बाकी के दोनों लोग आंसर नहीं करेंगे. कोयल, थैंक यू. Yes, the answer is C. Agarose gel electrophoresis. It is agarose gel electrophoresis. In northern blotting, a following probe is typically used to detect specific RNA molecules. What is that? In northern blotting, a dash probe is typically used to detect specific RNA molecules. Northern blotting हम किसके लिए करते हैं? RNA के लिए करते हैं. So the probe will be a cDNA, right? So the answer is DNA. Okay. Yes. Let's move towards the next question. Question number 4. Following enzyme is used to get a single cell suspension from the liver. Question number 4. Following enzyme is used to get a single cell suspension from the liver. Answer to question number 4, everyone. बताइए. देबाद्रता, कोयल, want to answer? So answer to question number 4 is collagenase. Okay, it is collagenase. Question number 5. Chloroform is actually going to remove the dash part. What part it is? It is the lipid part. Yes, it is lipid part. Question number 6. The phenol is going to denature the proteins and they will also become part of dash phase. Question number 6. The phenol is going to denature the protein and they will also become a part of dash phase. Organic. Yes, the answer is organic. Now let's move towards the next question. A smear on a gel is actually an indication that you have dash the genomic DNA, which suggests that you have either broken down or degraded the DNA, genomic DNA. So we have done what? Question number 7. Yes, it is B, it is fragmented. Okay, messenger RNA population can be isolated from RNA pool using dash affinity chromatography. Which affinity chromatography it is? Yes, it is poly T. Why poly T? Why only poly T? Question number 8 can answer. Why only poly T? Because of the presence of poly A tail at the 3' end. Okay. Next is the in northern blotting. To get a very good separation of these messenger RNA molecules, you need to run them on a denaturing dash gel. What type of gel? Which gel? Question number 9. Yes, it is agarose gel. Yes, right. RNA samples and agarose gel contain dash to denature the secondary structure present in the RNA. What is that? Question number 10 का answer. Yes, it is formaldehyde. ठीक है? It is formaldehyde. Next question. Dash is used for the preparation of RNA's free water. Question number 11 का answer everyone. Yes, डेप्सी. इसकी full form क्या होती है? Everyone, what is the full form of DEPSI? It is diethyl pyrocarbonate. Okay, लिखी भी हुई है वहां notes में. Next is the bromophenol blue is the dash dye used to monitor the progress of gel electrophoresis. What kind of a dye it is bromophenol blue? Question number 12 everyone. Bhavna, are you very sure it is a staining dye? स्टेन हम यहाँ कर भी रहे हैं कुछ. स्टेनिंग तो वहां होती है ना जब हम एसडीएस पेज चला रहे हैं. That too with Comasi Brilliant Blue. So bromophenol blue and Comasi Brilliant Blue are two different dyes. So what type of a dye it is? It is a tracking dye. We are tracking our samples on the gel na, using this dye. So it is a tracking dye. So answer to the question number 12 is C, tracking. Question number 13. After dissolving the gel, a thin layer of organic solvent such as butanol or isopropanol is layered to stop the entry of dash. What is that? Question number 13, everyone. Question number 13. Is there anyone who wants to answer or not? So question number 13 answer is oxygen. Okay. It is oxygen. Yes, Devadrita, you are right. Next question is ammonium persulfate is in the presence of dash form oxygen free radicals and induces the polymerization of acrylamide monomer to form a linear polymer. So in the presence of what? Yes, in the presence of timid, because timid is what? It is a catalyst. Okay, so it is a catalyst. Next is northern blotting is used to analyze the dash activity of the gene. Northern blotting is used to analyze the dash activity of the gene, and the answer is Yes, it is the translational. 2 मिनट. Northern blotting RNA translational कैसे? Northern blotting पे तो we are studying the RNA and that too particularly mRNA. So the answer to this question is transcriptional, not translational. It is transcriptional. Yes, it is transcriptional. So this with this we come to an end of the questions from your assignment also. Still, if you have any questions to ask, you can ask me. Any confusion, you can ask me. Any other questions, class? You can ask me if you have any questions. समझ में आ रहा है आप लोगों को ये टेक्निक्स जो हैं, नॉर्दर्न ब्लॉटिंग, सदर्न ब्लॉटिंग, वेस्टर्न ब्लॉटिंग, because these are the techniques. If you will think of making your career in molecular biology further, if any of you are students, then it will be very helpful for you guys. Guys must understand these techniques. What is the purpose of water blank in PCR analysis? So basically, water blank in PCR analysis tells you that whether the amplification is because of your specific gene, or was there something in the environment or some contamination in your sample, because of that the amplification has come. So that is why water blank is used in case of PCR analysis. Okay, Bhavna, did you get the answer? Perfect, thank you. Any more questions, anyone? No more questions? You understood well the southern blotting, the western blotting, the northern blotting? Acha, why do we do eastern blotting? Why do we do eastern blotting? I told you guys in the starting of the lecture. Tell me, why do we do eastern blotting? We haven't discussed eastern blotting, but I told you guys in the starting of the lecture. So why do we do eastern blotting? Tell me. Anyone? No one wants to answer. Yes, Bhavna. Detect post-translational modifications. Correct. So basically any type of carbohydrate, because in post-translational modification what happens? Most of the cases it is any sugar moiety is that is getting attached to the protein. So for that, to study those post-translational modifications, we generally do eastern blotting to study the carbohydrate. Okay, so eastern blotting is used for that. And apart from that, any other questions if anyone is having? Any other question? No questions? ठीक है, if you don't have any questions, you guys can leave the meeting. Thank you so much for joining. I'll meet you guys next Tuesday. You're welcome, Debadrita. You're welcome, Koyal.